Host-pathogen interaction. Host-pathogen interaction is the relationship between a host organism and a microbe or virus that lives within it. The interaction can occur at molecular, cellular, or organismic level. Understanding the host-pathogen interaction can help develop new treatments for infectious diseases. The first question that comes in our mind is that how does this interaction happens? As the host encounters the pathogen, the host's innate immune system responds to it. The host produces molecules like cytokines, chemokines to fight the pathogen. The host immune system also activates the T cells, which are involved in more specific immune responses. The second question that comes in our mind is that how does the host defend itself? The host has surveillance systems to detect the invading microbes, and the host has effector molecules that destroy the invading microbes. The third question is how does the pathogen evade the host's defenses? The pathogen has evolved strategies to evade or overcome the host's defenses. Pathogens enter host cells through the respiratory, digestive, genitourinary tracts or skin and are recognized by the innate immune system. The portal of entry is how a pathogen enters a host. The portal of entry must provide access to the tissues where the pathogen can multiply. The innate immune system recognizes the microorganisms that invade a vertebrate hosts. The innate immune system uses germline encoded pattern recognition receptors or the PRRs to recognize microbial components. Toll-like receptors and cytoplasmic receptors are examples of PRR that recognize distinct microbial components. Host sends glycoproteins of pathogens to induce resistance. Pathogen recognition by innate immune system. When a pathogen enters the body, the cells in the blood and lymph detect the specific pathogen-associated molecular patterns on the pathogen surface. These are also known as PAMPs. PAMPs are carbohydrate, polypeptide, and nucleic acids signatures that are expressed by viruses, bacteria, and parasites, but which differ from the molecules on host cells. These PAMPs allow the immune system to recognize self from the other, so not to destroy the host. The immune system has specific cells with receptors that recognize these PAMPs. A macrophage, which is a large phagocytic cell that engulfs foreign particles and pathogens, recognizes the PAMPs via complementary pattern recognition receptors or PRRs. The PRRs are molecules on macrophages and dendritic cells which are in contact with the external environment and can thus recognize the PAMPs when present. A monocyte, a type of a leukocyte, which is WBC, that circulates in the blood and lymph, differentiate into macrophages after it moves into the infected tissue. The dendritic cells bind molecular signatures of pathogens, promoting pathogen engulfment and destruction. Once a pathogen is detected, the immune system must also track whether it is replicating intracellularly or extracellularly. The innate immune system must respond accordingly by identifying the extracellular pathogen or by identifying host cells that have already been infected. Cytokine release effect. The binding of PRRs and PAMPs triggers the release of cytokines, which signal that a pathogen is present and needs to be destroyed, along with any infected cells. The first question that comes in our mind is what is a cytokine? A cytokine is a chemical messenger that regulates cell differentiation, proliferation, and gene expression to affect immune responses. At least 40 types of cytokines exist in humans that differ in terms of cell type that produces them, the cell type that responds to them, and the changes they produce. Subclasses of cytokines are interleukin, which mediates interaction between leukocytes. Interleukins are involved in bridging the innate and adaptive immune responses. In addition to being released from the cells after PAMP recognition, cytokines are released by the infected cells, which bind to nearby uninfected cells, inducing those cells to release cytokines, resulting in a cytokine burst. Interferons that are released by infected cells as a warning to nearby uninfected cells is the second subclass of cytokines. The function of interferons is to inhibit viral replication, making them particularly effective against viruses. They also have other important functions such as tumor surveillance. Interferons work by signaling neighboring uninfected cells to destroy RNA and reduce protein synthesis, signaling neighboring infected cells to undergo apoptosis, that is, programmed cell death, and activating immune cells. Cytokines are responsible for bringing the symptoms of sickness and increasing the core body temperature, causing a fever, which causes the liver to withhold iron from the blood. Without iron, certain pathogens, like some bacteria, are unable to replicate. This is also known as nutritional immunity. This diagram here explains the innate immune response to cuts. In response to a cut, mast cells secrete histamines that cause the nearby capillaries to dilate. Neutrophils and monocytes leave the capillaries, and monocytes mature into macrophages. The neutrophils, dendritic cells, and macrophages release chemicals to stimulate the inflammatory response. Neutrophils and macrophages also consume the invading bacteria by phagocytosis. Recognition of pathogen by adaptive immune system. In recognition of pathogen by adaptive immune system, two terms are of extreme importance. The first one is antigen and the other one is antigen receptors. Antigen is a molecule of often a protein that triggers an immune response when it is recognized as foreign, leading to the production of antibodies to neutralize or eliminate it. Antigen receptors are proteins on the surface of B and T cells that recognize and bind to specific antigens, initiating an immune response. T cell uses T cell receptors, TCRs, while B cell use surface immunoglobulins as their antigen receptors. Adaptive immune recognition is mediated by antigen receptors. The genes encoding these receptors are assembled from gene segments in the germline, and somatic recombination of these segments enables the generation of a diverse repertoire of receptors with random but narrow specificities. Antigen receptors are clonally distributed on T and B lymphocytes, which allows clonal selection of pathogen-specific receptors and is the basis for immunological memory. That is, each lymphocyte expresses antigen receptors of a single specificity. So, only specific populations of lymphocytes are selected to expand in the response to a pathogen. Thanks for watching. Kindly like and subscribe.